Now let's study regulation or control of glycolysis. So this pathway, glycolytic pathway, is controlled or regulated by an enzyme PFK, phosphofructokinase. So this enzyme phosphofructokinase is also called as pacemaker enzyme of glycolysis, which is involved in controlling or regulating the pathway. This is glycolytic, that is glycolytic pathway. Okay, let's see about this enzyme. So this enzyme consists of two sites. They are regulatory site and substrate binding site. This enzyme called phosphofructokinase. So this is phosphofructokinase enzyme. This enzyme consists of two sites. They are regulatory site and substrate binding site. So at this substrate binding site, substrate comes and binds. What is substrate for this enzyme? Fructose 6-phosphate is the substrate for this enzyme. So fructose 6-phosphate comes and binds with the substrate binding site of this enzyme. At regulatory site, an inhibitor or enhancer. Either an inhibitor or enhancer comes and binds. Enhancer. So what is inhibitor here? What is the inhibitor of this enzyme? Inhibitor of this enzyme is ATP. ATP and even citric acid. These two are the inhibitors of this enzyme. Whereas enhancer is ADP. ADP is the enhancer of this enzyme. Okay. Now let's see what happens when this inhibitor comes and binds. So inhibitor, that is ATP, has come and it has bound to the regulatory site of this enzyme. When it has bound to the regulatory site of the enzyme, it results in change of the shape of the substrate binding site of this enzyme. It results in change of the shape of the substrate binding site. So this is PFK enzyme. So here there is change in the shape of substrate binding site. So if there is change in the shape is changed. When there is change in the shape of substrate binding site, what happens? The substrate cannot fit into this substrate binding site of the enzyme properly, and hence affinity of the enzyme towards substrate. Affinity of the enzyme towards its substrate will be decreased. When the affinity of the enzyme towards its substrate will be decreased, what happens? The activity of this enzyme will be decreased. So when the activity of this enzyme is decreased, what happens? Rate of glycolysis will be decreased. So glycolytic rate will be decreased. So like this enzyme phosphofructokinase is involved in inhibiting glycolysis, controlling, regulating the glycolysis. Now let's see about enhancer. ADP is enhancer. So actually when cell is rich in ATP, cell doesn't need no more ATP. When cell doesn't need no more ATP, that ATP comes and binds with the with the regulatory site of this enzyme. When cell is in need of ATP, then what happens? Enhancer, that is ADP. When cell is in need of ATP, that cell will be ADP rich. So that ADP comes and binds with the regulatory site of this enzyme. So when ADP comes and binds with the regulatory site of this enzyme, there will be no change in the shape of the substrate binding site of this enzyme. When there is no change in its shape, what happens? Affinity of the enzyme towards substrate will be same. It will not be decreased. When it is not decreased, the activity of the enzyme will not be decreased. It will be same. And glycolytic rate will be enhanced. So what happens? So glycolysis will be proceeded. So this type of inhibition, this type of regulation by this enzyme is this type of inhibition of the enzyme is called as allosteric inhibition. Allosteric inhibition. Hope you understood this concept, students. Thank you so much.